My name is Natalie, and I'm a director at the Woodsworth College Alumni Association. For those of you joining online, we have enabled live captioning, so you can view the captions by selecting the CC button at the bar at the bottom of the screen and selecting Show Captions. We would like to begin by acknowledging the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work and gather on this land. On the agenda for tonight, we will hear a lecture from Dr. Shaf Keshavjee titled The Future of Transplantation, Engineering and Distributing Superorgans. A Q&A session will follow the lecture. For our audience online, please submit your questions and comments in the Q&A tab on your Zoom screen, and we will include them. Dr. Keshavjee is a thoracic surgeon and director of the Toronto Lung Transplant Program. He is Chief of Innovation at the University Health Network, or UHN. He is Professor of Thoracic Surgery and Biomedical Engineering and Vice Chair for Innovation, Department of Surgery at the Temerty Faculty of Medicine. Dr. Keshavjee completed his medical training at the University of Toronto and then had training at Harvard University and the University of London for airway surgery and heart-lung transplantation, respectively. He received the Lifetime Achievement Award from the Canadian Society for Transplantation and the American Society of Transplantation Innovation Award for contributions to the field of lung transplantation. He also received the FNG Star Award in 2021 for the highest national award from the Canadian Medical Association, recognizing a physician for their outstanding and inspiring lifetime achievement. He is a fellow of the Canadian Academy of Health Sciences and has been awarded honorary Doctor of Science degrees from both Ryerson and Queen's Universities. He was also awarded an honorary fellowship to the Royal College of Surgeons of Edinburgh for his service to mankind. Dr. Keshavjee has received 2 Queen Elizabeth II Diamond Jubilee Medals and the Governor's General Award for Innovation. He was awarded the Order of Ontario in 2014 and was appointed the Officer of the Order of Canada in 2013. We are honored to have you here, and now I will pass it over to you. Thank you.
Thanks very much for that kind introduction, and it's an honor to speak here as an alumnus of the University of Toronto. So I'm going to talk to you about where we're going in transplantation and the future that we're inventing. And I think it's a really important thing for you to know that this is all work done here in Toronto that we lead the world with. I have a number of relationships to disclose. There are 3 companies that we've spun out related to the work and the research that we've done. And we have a number of industry partners that have helped us and we continue to collaborate with in doing this work that we definitely could not do alone. So when you think about organ preservation for transplantation, the idea that you could take an organ out of someone who's died and transport it and put it into someone else is quite remarkable and miraculous. And the way we achieved that was with cold flush preservation. That's been the cornerstone of organ preservation. So you flush the organ with a cold solution, it cools it down, and basically the conceptual focus is that you slow down the dying process. And although it's protective, it hinders the possibility of other metabolic processes because it shuts down the organ and shuts down repair as well. And so that it also hinders the ability of the organ to repair and regenerate itself. And really importantly, you only find out how the organ's going to work after you put it in and take the clamps off. And that makes us very conservative as doctors, like you're not going to take an organ that you don't think is going to work. So when you look around the world and in Canada and the United States, only 20% of lungs that are donated actually get transplanted. So even if you sign your donor card, only 20% are felt to be suitable. And we make that decision in the operating room and the organ doesn't look or doesn't meet criteria, we don't transplant it. And even when we take the perfect ones that we think are okay, 20 to 30% of the time we have what's called primary graft dysfunction, and the organ doesn't work as well, and the patient's at risk. So the first paradigm shift we considered was, if we took the organ out of the body and put it on a machine where we could evaluate it, maybe we could use more organs. And I think that we could triple the number of transplants that we could perform. And we've shown that already, that taking 50 to 60% of lungs to transplantation. But if you take a lung outside the body and you find out that it's not okay, you don't want to look for reasons not to transplant them, but if it's not okay, can you fix it? And if you fix it, can you interrogate it and show that it's fixed, that now you can transplant it safely? And I think if we get to this point, we'll use all the organs available to us and decline very few. And the whole idea is using this diagnostic and therapeutic or precision medicine approach to not just keep an organ the way we found it and make sure that it doesn't deteriorate, but actually, can you make it better than the way we found it? Can we prepare an organ for that unusual journey of transplant that we're going to expect that organ to undertake? So many years ago, I got interested in interleukin-10. Interleukin-10 is a sign, a message that cells use to communicate with each other. And it's a really key cytokine in transplants, in that what it does is it downregulates the innate immune system or the inflammatory system that is turned on when an organ has blood returned to it. And it also turns down the adaptive immune system, which is the one you think of in transplant, the rejection system. So it makes the T cells, instead of being attacking T cells, makes them the more regulatory or accepting or tolerant T cells. So you can see that IL-10 in a transplanted organ could be very useful to have. And the idea we had some years ago was, can we actually make the organ make more IL-10 so it's more likely to be accepted? So we took the common cold virus, took the virus genes out of it, put in the IL-10 gene, used the virus to get into the lung, and the lung viruses are very good at getting into the lung, as you know. It carries the IL-10 gene in, and then the cells make IL-10 locally. So you basically have an organ that its own immunosuppression factory there. And we showed that that works. In this experiment here on the left are rat lungs stored for 24 hours and then transplanted. And then in the green here, you'll see if they had IL-10, after you transplant them, they work better. And we did the same experiment in pigs on the right-hand side there, saying that if you transfect the donor lungs with IL-10, upregulate the IL-10 gene, you can get those lungs to work better after transplant in pig experiments. A very important proof of concept that you can modify the donor to work better in the recipient. And then we realized, well, that wasn't entirely practical to have to go work on the donor. It takes about 6 or 9 hours for the gene to upregulate, then take the organs and then transplant them. So we said, could we create a system to have the lung outside the body and still have the gene therapy? Now, remember, I told you when you cut it down to 4 degrees Celsius, everything shuts down. It's gone to sleep. So it's not going to undergo gene therapy either. So we had to bring it back up to 37 degrees, normal body temperature, in order to do gene therapy outside the body. So we created the Toronto ex vivo lung perfusion system, another breakthrough, where we put the lungs in a chamber. You ventilate it with an ICU ventilator, as good as any ventilator you would have a patient on in the intensive care unit. And you sweep it with a gas, which actually makes it as if it's running backwards. It removes oxygen from the system and adds carbon dioxide, so you can then measure the lung's ability to be a lung, to add oxygen and remove carbon dioxide. And we used all of the parameters that we've learned on how to protect the lung, to not injure it and keep it outside the body. This was our original component system that we introduced, and we reduced it to a machine that you see in the bottom right there that is now commercially available for other centers to do what we've been doing. The key experiment, Marcelo Sippel was my fellow at the time and currently my colleague. What he did is he put normal pig lungs on our EVLP system and kept them for 12 hours. So this is a pig lung outside the body at 37 degrees. And you can see the lung function was excellent for the entire 12 hours. And not only that, we showed that you didn't need oxygen, you didn't need red cells, that you could get the same function without red cells, which was controversial at the time. And most importantly, after 12 hours at 37 degrees, these lungs functioned almost normal. That was unheard of. That was a major breakthrough in the field in 2008. You could take lungs outside of the body, have no deterioration, and show that you can improve them after function. So now we had a platform where we could repair lungs outside the body. We then did a very important experiment looking down the road to how will we translate this clinically. We took pig lungs, we stored them for 12 hours in cold, the way we normally do clinically. And for the second 12 hours, we put them on normothermic ex vivo lung perfusion for a total of 24 hours of storage and then transplanted them. And we were looking for equivalence, but in fact, the lungs that had been on the EVLP were significantly better, which we understand better today than we did at that time. The important thing here is we now have a paradigm where we can pick up a lung anywhere in North America. We go to Vancouver, we go to LA, we go to Halifax, anywhere we want to go, pick up a lung, bring it to Toronto, put it on ex vivo perfusion, and evaluate it and transplant it. So now what we've got is we've got the ability to manipulate the temperature of the organ. When you want it to go to sleep and protect it while we're sewing it in, while we're taking it out, you can cool it. When you want to actually work on it or evaluate it, you bring it up to normal temperature. So it's improved utilization. It's improved our ability to treat the organ like you would treat a patient in a personalized medicine fashion. It's allowed us to create organs with a predictable outcome. We know that that lung's okay and it's going to function well. It's improved the safety of transplant. And for those of you that haven't seen it on YouTube yet, this is what it looks like, where Grey's Anatomy has had it as well. These are human lungs. They were taken out of the body about 9 hours before you see them here. They're breathing, they're being perfused. This is a fiber optic bronchoscope, so you can actually look inside the lung. You can sample and you can deliver drugs. And here we're delivering the gene therapy that I talked about. And then over time, you can continue to evaluate the lungs and see, are they getting better or not? If they're getting better, go ahead and transplant them. So this was a complete transformation. And there's Marcelo Sippel and John Young, who did his PhD with me as well, in the operating room when we brought the first case clinically in the world in 2008. And as I said, that was our component system, which we've now made into a device that does it a lot simpler and a lot more easier. We published in 2011 in the New England Journal showing that we could take lungs that we wouldn't use, that no one would use, improve them, and have equivalent outcomes after transplant. When we'd done over 300 other cases, we showed that the non-EVLP or standard lungs in blue performed exactly the same as lungs that went on EVLP in green, showing that we could salvage lungs and have equivalent or even slightly better outcomes with ex vivo lung perfusion. So what has this done to our program? Many of you may not know that the first successful lung transplant in the world was done right here at Toronto General Hospital in 1983. The first double lung transplant in the world was done in 1986, here also at Toronto General. And over the 90s and early 2000s, we really pushed the boundaries in using lungs that weren't perfect. We're called marginal or extended donors, doing our best to use the lungs we could get hold of and get patients through a transplant, albeit at a riskier rate. And in the mid-2000s, we kind of plateaued at about 100 transplants a year. In the EVLP era, we fully doubled the number of transplants that we perform to over 200 a year now at Toronto General, and it's still the largest lung transplant program in the world. Here you can see that in blue are the lungs that go straight to transplant. In yellow are the lungs we rescued and treated on EVLP and took to transplant. This is probably one of the most important slides that I can show you about the impact of this research. In red is the operative mortality of lung transplantation in our program. In the 80s, you did have a 50-50 chance of getting out of the operating room. It was a very risky operation. It was miraculous, but it was risky. As we pushed the limits, you can see we had ups and downs in our operative mortality as we would push the boundaries. But now you can see we're over 200 transplants a year, and our operative mortality is only 1.5%. So we've got more lungs, saving more lives with a safer outcome. And this is what we need to do in all of organ transplantation. Most people around the world think that an organ can only be preserved for about 6 hours or so in lungs and hearts and so on, and they don't tend to go over 6 hours. And what we showed, John Young published this in the Lancet, we looked at lungs that were over 12 hours preservation outside the body and compared greater than 12 hours to less than 12 hours. And you can see that for lungs on the right-hand side, in green are the ones that were preserved more than 12 hours, in blue are the ones less than 12 hours, and really they're equivalent in our program. Importantly, when you look at the 90-day mortality, there were only 2 deaths, and you can see the deaths are not at the far extent of the preservation time. So really, we have shown the world that you can go further than 12 hours and get lungs. And what this has done is really improved the logistics of transplantation. And this is the emergency board at Toronto General Hospital on one day in June of 2015. And you can see there's a laparotomy, a kidney transplant, a liver transplant, a double lung kidney, double lung, heart transplant, lung transplant, double lung, liver transplant, and a double lung transplant. Like we did 5 lung transplants in just about 30 hours. And these are operations that take about 12 hours. So how we do it is the first lung went on ex vivo perfusion. We performed 2 single lungs. The second one went directly to transplant. The third one was checked on ex vivo perfusion. It didn't work. The fourth one was put on ex vivo just to wait until we had an operating room and a surgeon available, and therefore we can deal with that. I think it's really important to look at the future of transplantation and think about where is this field going? How do we scale it? How do we sustain it? People don't like working in the middle of the night. We're not attracting the surgeons of the future into this very tough specialty where you start your operation at 2 in the morning and you go for 12 or 14 hours. And there's been work showing that the outcomes of lung transplant done at night in green here are not quite as good as the ones done in the daytime. So there are circadian rhythms that humans work on, and even ourselves do that, and we do better at normal times. So can we change how we do transplant and schedule transplant? So what we did is we looked at a study of avoiding overnight transplantation using 10-degree storage, because we'd done some work showing that if you store lungs at 10 instead of 4 degrees, you have better mitochondrial protection, better mitochondrial preservation. Mitochondria are the little energy-creating organelles inside your cells. So we took lungs and said, if you cross-clamp after 6 p.m. at night, you won't start your operation until 6 a.m. So you have 12 hours delay to do it. In the pilot study, you can see, again, this is the donor x-rays, and all of these x-rays on the right are the admission to the ICU 24, 48, and 72 hours after transplant. Just looking at it even as non-medical people, you'll see nice black, well-aerated x-rays. All these patients did very well, and the preservation times were ranging from 10 to 15 hours for the first lung, and from 11 to 16 hours for the second lung, with no primary graft dysfunction, a hospital length of stay of 17 days, which is the same or a little better than our usual, and 100% 30-day survival. So showing that this new preservation technique can actually do that. We then went on to do a larger clinical trial of 70 patients. And again, with preservation times going up to 17 and 19 hours, had excellent lung function. And we're currently taking this through a multi-center international trial to validate the concept that lung transplantation can be a scheduled operation rather than a middle of the night emergency. And you can see the 30-day survival of the 10-degree ones is 100% versus 96.4% in the control. So certainly comparable results. And this is the 1-year survival again, 10 degrees versus the matched controls in our program. So again, we've changed it so that you don't have to be doing transplants in the middle of the night. The other part we looked at, though, is can we actually use the combination of the 10-degree preservation with the ex vivo lung perfusion? And so Adil Ali, who's a PhD student with Marcelo, did this work where he took lungs, stored them for 6 hours at 4 degrees, so just bring them from anywhere in Canada to Toronto, then 18 hours at 10 degrees, 4 hours of 37 EVLP, 20 hours at 10, 4 hours, 20 hours, and then a transplant. So really 3 days, those lungs, before we transplanted them. And you see excellent lung function after performance, after transplant, and saying that now if I want to work at gene therapy, cell therapy, CRISPR gene modification, I have 3 days to work with those lungs before we transplant them, which is a remarkable opportunity. So we've really defined the state of the art and how you preserve lungs, and continue to teach clinicians all over the world this way. But the exciting opportunity is now that you have this technology, what can you do with lungs? And just like you have personalized medicine for patients, you can have personalized medicine for the organ. We're looking at perfusion. I've been very interested in cell therapy and gene therapy. Others are looking at immunocloaking, other gases, other pharmaceutical agents to repair lungs outside the body. And I think that it's important to take note of the fact that all of the precision diagnostics that have been developed for clinical medicine are developed for chronic diseases or cancer, where, if you get your gene profile tomorrow, that's fine. But tomorrow is not fine for transplant. We need an answer in 10 minutes. We need an answer in 30 minutes. So we've been working with a number of companies to develop tests that we can interrogate the lung and get an answer right away. So we know, one, it's better. If it's not better, what the fix of that lung should be. And we have the Toronto Lung Score that will tell you that when this lung is on ex vivo perfusion, you can predict that your patient will be fine, that they'll be off the ventilator in 3 days. That's a good lung, go ahead. Or it's not a good lung, you've got problems, and your patient's going to have a difficult time. Again, we don't use mortality as a test for these things, because it's so rare to have a death after a lung transplant now, thankfully. But how are you going to say that, can you transplant another lung that you normally wouldn't do? You look at the probability that your patient's going to be off the ventilator in a few days. And that's what our Toronto Lung Score has done. And we're working on developing that to make that available to everybody. I mean, on ex vivo lung perfusion, if lungs don't have good oxygenation, we put them on, we clear out the fluid, and we transplant the lungs. Patient arrives in the ICU with excellent lung function. We had a donor that had clots in the lung artery, in the pulmonary artery. We treated the patient with alteplase, like you would treat a pulmonary embolism or a heart attack or a stroke in a patient. We monitored the result over time, saw the pulmonary artery pressure come down. We took a biopsy of the lung. The pathologist said there was no signs of chronic pulmonary hypertension, and we successfully transplanted that lung, and the patient was off the ventilator in a day. So these are lungs we never would have used, but we actually treated them with drugs we currently have on the shelf. The second most common reason lungs are not used for transplantation is the donors in the ICU on a ventilator, they get a pneumonia, they get infection in the lung. So the question is, when can we treat those lungs? Will those lungs be sterile and safe enough to transplant? Can we treat the infection outside the body? When the lung is by itself in a machine, There's no kidneys, there's no liver, there's no eyes or ears for toxicity. You can use very high dose antibiotics to sterilize the organ, and we're working at that. We were the first in the world to safely transplant lungs from a donor with hepatitis C into non-hepatitis C recipients, and showed that we could do that with treating it. But the question is, most patients will seroconvert, and there is a drug for hepatitis C. It's very expensive, but it's treatable. And so many patients are alive today because of our work, that you can treat them with the antiviral drugs after transplant. But what we've been looking at is, can we use ultraviolet light to sterilize the organ before you transplant it, so that the virus would never be transmitted with the transplant. And for other viruses like HIV, like hepatitis B, and maybe some of the porcine xenoviruses from animals, maybe we could learn to completely sterilize an organ so we never transport a virus with it. And in this paper that we published in The Lancet, out of 9 patients, 2, the organ was completely sterilized with this technique, where the recipient never seroconverted. Now, 2 out of 9 is not there yet, but we'll get there, and I think those are the important things. There are many patients who are blood type O, and they're small, and they wait a long time for a lung. And so what Marcelo's been doing is looking at, can we change the blood type of the organ? So we put them on EVLP, and he's been using an an enzyme, to remove the type A and B antigens on the lung to make a universal donor organ. So if you have a type O organ, just put it in whoever needs it the most and whoever's the right size. So we're working on these sorts of opportunities, and this data just basically showed that he cut the A enzyme down to less than 3%, which would be tolerable for a transplant. So I told you about the gene therapy. I was using adenovirus to take the virus vector into the cells and have the lung make its own IL-10. There's some issues with adenoviral vectors, like they will insert a gene. First of all, you got to carry the whole gene. Secondly, it'll insert it in a sort of random fashion. So sometimes there could be errors or unintended consequences of that randomness. And also it takes time. So John Young did the work showing that if you use the adenoviral vector on ex vivo, the purple squares on that side shows that you have the IL-10 level exactly the same as if we treated it in the donor. So we don't have to run all the way to Vancouver to put our vector in to the donor. We can bring the lung home, do it here on EVLP. We did, Tiago Machuca, who's now running the lung transplant program in Miami, did these pig survival studies where we transplanted pigs and had them survive, and showed that at 7 days, these pigs were not rejecting the lung that we transplanted them when we had treated them with the adenoviral IL-10 gene therapy. Marcelo did this work on human lungs that were not used for transplant, that if you treated them with the IL-10, going from the red to the blue, you could improve the oxygenation, decrease the pulmonary vascular resistance, and bring these lungs to transplantable quality that we wouldn't have transplanted. And when we looked at the injury to the lung, we showed that the IL-10 gene therapy was turning around the inflammatory profile of the lungs from brain death injury. So again, making better organs for transplant. This is a very neat picture. This rat here with the rainbow picture is bioluminescence imaging. And what we did is we treated the rat with the IL-10, this was lenti-IL-10 actually, a different virus, at the time of transplant. And what we showed is that organ expresses the transgene for a full year after one treatment at the time of transplant. So you truly have created an organ that immunosuppresses for itself. Fast forward to the current era, CRISPR was invented and discovered. And I think CRISPR is probably the most significant discovery since the discovery of DNA itself. When you think of what I was doing with ad vectors and lentivectors and random insertion of vectors into your genome, CRISPR is like a word editing system. It will put the gene exactly where you want it. You cut one piece out and put what you want there. And more importantly, we found that we can, instead of having to carry IL-10 in, we can just turn IL-10 on. So with CRISPR technology. So we're working on that. Kumi Misaki from Japan is working in my lab doing her PhD, and basically has developed the guide RNAs and the constructs to use CRISPR to whole organ gene edit. And that is a major breakthrough. If we can do that in larger organs, it will be quite amazing that you can permanently and safely and precisely edit an organ to do what it's supposed to do for a transplant. We're also looking at, can you put stem cells into the organs? So here again, the gene therapy, CRISPR editing. On the other side, cells. Can you put cells in the stem cells that can repair lungs, or immunologic cells to immunomodulate the lungs before you introduce it to the recipient? And so we did the study showing that if you put these cells into the EVLP circuit, they do get stuck in the lung. The green dots are the cells lodged in the lung, so they do get there. They're not stuck in the membrane or the tubing and the circuits. We can engineer cells so that they are not just plain stem cells, but supercharged, if you will, with the IL-10 gene or other things, and then put them into the circuit. We also recognized in this study, Antti Nikkanen from Finland did this in our lab, that if you put these stem cells into very injured lungs, you're putting them into a very hostile environment where even they can't work as well. So if you want to use these neat biologic therapies, you've got to put them and create the environment ex vivo that will create an opportunity for success of these unique therapies. Stephen Juvet, who's one of the respirologists in our team, is doing a very cool set of studies. So this is what we intend to do. So what you do is you take the recipient and you take their regulatory T cells. These are the cells that are the tolerance generating T cells that will accept the organ. You amplify them outside and you store them frozen. At the time of the transplant, you put the lungs on ex vivo perfusion and you put your own regulatory T cells into that organ and then transplant it. So you're introducing the organ to the host in the context of self. We have never had that opportunity for tolerance. So this is a completely unique angle that this has created for us to be able to create a Treg permissive immunosuppression strategy for accepting the organ. I mean, you got to understand that transplant is fantastic. You do a transplant, you save a life, and then that person spends the rest of their life trying to destroy that organ. So how can you tell the body that it's me, itself? And I think this is a unique opportunity. In our lab, we have about 130 people at UHN, work at the Toronto General Research Institute working on this. We do screening studies in cells, small animal, large animal studies, and then pig ex vivo lung perfusion and transplants, and then human ex vivo lung perfusion. And studying lungs that donors donate for as organ donors but are damaged and can't be used for transplant are very valuable, because we can take those damaged lungs to the lab and study, can we fix those lungs? And we've had many categories of lungs where we've learned, yes, we can fix this. Next time we take it straight to the clinical OR rather than to the research lab. We're taking what we've learned in lungs to help the other organs, the heart, the lung, the liver, the pancreas teams. So in our lab at Toronto General, here's the ex vivo lung machine. And then we have other teams working on the other organs as well. So at the same time, the ex vivo heart. So they're learning, how can we keep a heart outside the body as well? And then we've got the liver team working on liver systems as well, in terms of can we take these same concepts to repairing organs, keeping them alive longer for all our organ systems in our transplant program. And then more recently, even the pancreas, which has been a difficult one to manage and transplant. So that's sort of a unique opportunity for ours. So when you think of the things that we can do to organs now, how do we achieve this whole vision? And I think, most people say, well, transplant is already so complex as it is. How do you add all of this to it? You're adding more advanced therapy, you're adding more diagnostics, you're adding more complexity, you're adding devices, you're adding cost, you need staff. And I think the whole system will have to change: organ allocation, transport, and distribution. And I think I'd like you to think about this, that the optimization of organ management will be in specialized organ centers. And we can take a lesson from the evolution of blood transfusion. The first blood transfusion ever was from a healthy soldier to a hemorrhaging soldier in a battlefield. And you can imagine that they probably saved a few lives, but they probably killed a few people with blood transfusion reactions and infections. So then we developed MASH units, where we had units of blood in a fridge out in the field, and you'd give blood to people when they needed it. Again, room for errors, storage problems, distribution, availability. So we developed standardized, centralized collection centers, where you would go give your blood, it's taken out, it's controlled for quality, standard operating procedures, infection control, shelf life, inventory, tracking, distribution, and safety standards. And more importantly, for a very scarce resource, like a unit of blood, it's separated. So people that need cells get the cells, people who need cryo get the cryo, people who need the platelets get the platelets, and you can make a scarce resource go further. So it's allowed us to scale up, achieve cost and utilization efficiencies. I think we need to go the same way with organ transplantation. And I would argue that in organ transplantation, we're still at the mass unit stage of how we manage organs. Can we apply these concepts? I think this is the future of transplantation. It's going to be, we retrieve organs, we bring them to an organ repair center, we optimize them, and we ship them out to the recipient center, optimized for the particular recipient. And this might sound like science fiction to you, but we did build the first organ repair center in the world at Toronto General Hospital in 2008. There's our lung system, our liver system. Others are working on heart and kidney. And the Organ Regeneration Lab, OR18, at Toronto General was the first one. We now have 3 of them at Toronto General. And that was the original one where the lung was sitting on a table, perfused by the pump, and monitored by another monitor, and had a ventilator hooked up to it. We partnered with United Therapeutics, and we built the first lung hospital in the world. This is a 28,000 square foot facility in Silver Spring, Maryland, has 6 procedure rooms that can do ex vivo lung perfusion. And at full capacity, we could do 1,800 lungs a year in this facility alone. And these are what the rooms look like. These are organ perfusion specialists that are all trained at Toronto General for lung bioengineering. And they serve 32 hospitals in the US and Canada for providing lungs are brought in, repaired, and sent out again for transplant. You get all of the details on your iPhone. I can evaluate the lung. You can look in it. You can look at the oxygenation. You can get the pulmonary vascular resistance and the X-rays, images, and ventilation. And then you decide, is this a good lung? Am I going to transplant it or not? We built Lung Bioengineering 2 in Jacksonville, Florida, in partnership with the Mayo Clinic. This is the LB2 facility. LB3 is going to be in Phoenix, Arizona, and then LB4 here in Toronto finally. So that would be nice to see that come home. So really we're covering the whole continent with lung repair centers to be able to provide lungs to everyone that may need them. And when you think about it, we can't keep building whole operating rooms for one organ at a time, which is what we currently do. So we've reduced that entire room to one device so that the organ perfusion specialist can actually be looking after 10 lungs at a time instead of one lung at a time going forward. So When you look at the personalized medicine approach, lungs are in the donor hospital. Some go directly to transplant if they're fine. If they're not fine, go to ex vivo assessment, assess them for transplant, repair them. If they're not yet repaired, check them again, repair them. And when they're good, transplant them. If not, use our diagnostics to tell you that that lung is just not safe to transplant and you're not going to use it. Right now, the ecosystem is that you have a donor hospital, you have a recipient hospital, and we transport organs by land, by helicopter, by plane. Now you're going to see an organ repair center. So the organs will stop off at a repair center, get optimized, and then sent to the transplant hospital. And when you think about it, why would you send a Learjet to carry something that weighs less than 2 kilograms, right? And it's expensive, it's not good for the environment, and it's just not scalable and sustainable. So we've looked at ways to actually transport organs by drone, and here's a drone taking off from Toronto Western Hospital and flying across the Toronto skyline to Toronto General Hospital to deliver lungs for transplant. So, you know, you'll say, well, what's the big deal? You just flew across Toronto. The most difficult thing about flying a drone is flying in a populated city. There's radio frequency interference, so we built a GPS that was immune to that. You're flying over people's heads and you have to be safe. So we built a ballistic parachute. So if the drone fails, the parachute will fire and the drone will come down slowly to Earth. We now have drones that can go to Calgary and back and bring an organ. And we're working with Pearson Airport. We were just featured in Toronto Life, 9th reason to love Toronto. His organs are delivered by air. So that was 1 o'clock in the morning on the top of TGH. But you know, we're now working with Pearson Airport. The next challenge is flying in and out of commercial airspace. So we've created a drone port on the top of Toronto General and a drone port at the south end of Pearson Airport. And we're going to create a drone corridor from Pearson to TGH to be able to bring organs back and forth as the next step, and a very good use case for the development of drones. So I'd like to close by acknowledging the Latner Lab. We have, as I said, over 100 people working on all of this work that I've presented to you, and our clinical team that is probably the largest and most adventurous lung transplant team in the world that brings all of these innovations to the bedside. So I'll stop there and I'll be happy to take any questions from you.
Thank you very much for that very interesting talk. I will start off with questions from the floor. Are there any questions to start off with? Yes.
Thank you so much. First off, that's 100% amazing. Thank you so much for that talk. It was so interesting. I'm just curious about the systemic like integration of the organ into the person's body, because let's say like before the transplant, something was wrong with their organs. So they got a transplant, got a new organ, but what if their body is still not producing the right, I don't know, whatever systems to maintain it? So how does it integrate?
Yeah, you know, the issue is a lot of what we're learning from transplant and repairing organs will fit in the area of regenerative medicine. So if we can figure out how to prevent your lung or other organ getting injured and repair it in you, then maybe you'll never need a transplant. There are very few to none of the lung diseases that recur in the organ after it's transplanted. So, I mean, that's one thing that's good. You get another set of problems. So one is you get chronic rejection. So your body works to continually reject that organ. We give immunosuppression pills, but that suppresses your whole immune system. So you're more vulnerable to infection, you're more vulnerable to cancer, and you still get a form of rejection where the organ doesn't last forever. So the mantra in our lab is make a lung that'll last forever. So the idea is if you had the organ looking more like self, then you wouldn't have to give the systemic immunosuppression and make it safer. Yes.
So To add on to this concept of using T cells,
There's a mic there.
what has the success rate jumped in once you've used that technology? Because you've shown that curve.
Yeah.
And how old is it? This technique you're using right now?
Yeah. You know, we've been doing this for 10 years, ex vivo lung perfusion, but the therapeutics, I'm embarrassed to say, we haven't quite jumped with all those advanced therapeutics yet. So treatment with antibiotics, treatment with antivirals, treatment with the anti-clots, like drugs we have that are very routine, we use routinely. The T cell is really still animal studies. So we're just bringing those to the bedside now, you know, in terms of clinical trials. So the T cells really help rats not reject their lungs, but we don't know the answer in humans yet. Now, the early studies, because we can do ex vivo human lungs, the early studies, what Stephen Gervais is looking at is putting human lungs on EVLP, delivering human Tregs, and looking at the early interaction between the dendritic cells, which modify the immune response, and the regulatory T cells. And that all looks like our idea is correct. This is going to work. So I think we will move that clinically. The first clinical trial that I'd like to do is the gene-modified lungs. That'll be first in the world of gene-modified or gene-engineered organs for transplant. And then the Tregs are also lined up for that.
I think we have a question from online.
Yes.
The question is from Andrea. How many of these techniques would be used for other organs, such as heart, liver?
Yeah, the really important thing, you know, the lungs were the last of the major organs to be successfully transplanted because they're the hardest to do. And we kind of leapfrogged along ahead of everyone. Kidneys were the first transplanted, then hearts, then livers. These techniques are applicable. And I think while many people have dreamt about doing ex vivo perfusion, I mean, Da Vinci had drawings of ex vivo organ perfusion. Many people tried it. I think one of the challenges or the stray paths was thinking that one system would be fine for all organs. And what we did is we said, what does a lung need in the stressed and injured state? What is the best way to protect the lung in the stressed and injured state? And what is the best way to repair a lung? And I think now heart people and kidney people and liver people are looking at systems specifically for their organ. And I think doing it that way, Peter Friend and his group at Oxford have done in liver what we've done in lung pretty much, and they have an ex vivo liver system working. So I think these are generalizable, and that's what we're working with our group. And we have a New Frontiers grant from the CIHR that is really a grant to advance these concepts to the other organs.
Thank you very much. It was explained in a way that anybody who didn't have any medical knowledge could understand. So I appreciate that. And you touched on my question before in one of the earlier questions, and it's about going backwards. So if I'm the recipient, how can we avoid me being a recipient and have my heart improve? Now, obviously, the one with the high antibiotics is not feasible because of the other organs that are going to get affected. But what are some of the ways that it can be moved? Some of these techniques can be moved back to the recipient to avoid being a recipient and just having a better heart, a better lungs, better whatever. Thank you.
Thank you. So one point is that the ability to take a lung like this, we have learned so much about lungs that we couldn't have learned before, because every other system we studied lungs in was injuring it, and we were studying a form of injury rather than normal biology. So now that we can support a lung better, we can learn more about lungs, we can learn, and we're using EVLP systems to test new drugs. So for pulmonary fibrosis, like if we can test a drug on a human lung in EVLP, we will save the drug companies a decade of clinical trials. I mean, that's millions to billions of dollars. So I think that way, what we learn on these systems can do it. The other part of it is if we've learned how to perfuse and sustain the lung outside the body, can we do it in the body? And so we're looking at those kinds of repairs. The first one we're doing, we have an active clinical trial on that, is treating some cancers in the lung, colon cancer and sarcoma spread to the lung. As thoracic surgeons, sometimes you'll have a colon cancer removed. You'll have metastases to your lung that we take out as surgeons. And if you take out metastases from the colon in the lung, you have a 40% cure rate. So that's better than the 0 if you didn't take it out, but it's only 40%. And why? Because there's still some cancer in it. We're sometimes limited by the dose of cancer therapy that you can give, chemotherapy, because of toxicity to your eyes or kidney or whatever. So we've taken our ex vivo technique and doing it in vivo now. So we perfuse, excuse me, we perfuse the lung in you, isolate it from the rest of the body so you can put a high dose chemotherapy in the lung, get it out of the lung, and none of it goes into the patient. So that way you can actually use effective anti-cancer drugs that are toxic without having the toxicity go to the body. So that's one in the person. The other part is in adult respiratory distress syndrome. So COVID is one of them, and there's others from bacteria and viruses and so on. ARDS is really when your immune response to the virus or the other bacterial infection is sort of over exuberant, and the neutrophils and everything go on to create more injury. So others are looking at the concept of if you put those people on in vivo lung perfusion, can you dial down that inflammation? So I think, you know, those are some of the little forays into the recipient, but I think the strongest part about what you're getting at is, can we learn about lung regeneration? Can we figure out how to deliver stem cells to the lung and show that it's repairing the lung? If we can do that, every disease that you have is a failure of stem cells, right? If you get a lung injury, your lungs get better. Some people get a lung injury and they get pulmonary fibrosis. Why? Why do those lung cells go from lung cells to scar tissue? And it's because the regeneration pathway has gone awry. So I think learning those pathways will be the key to avoiding transplant in the future.
Okay, we have one more question online, if that's okay.
Okay, sure. The question is, what sort of changes do I foresee in the system to accommodate EVLP clinically, I presume? That's a very important question. And these are some of the things where we realized, like, we can come up with a really cool solution, but it needs to be incorporated into the workflow and the ecosystem of transplantation. And a very good example is the fact that in the United States, you can only take an organ out of a dead person if you have a named recipient, right? So if the lung is going to Mr. A, then you can take it. They charge the donor hospital the charges for that. They charge the recipient hospital for Mr. A and the organ and the transport and so on. But if you don't have a recipient, you cannot take the organ. What we saw is 80% of lungs are not being used, but nobody would take it because they don't meet criteria. We could take it and fix it, but we can't take it because nobody wants it yet and we don't have a Mr. A lined up. So we changed policy. We appealed for it and got UNOS to change the policy so that now in the United States, you can take a lung out, repair it, and then offer it. And that's increased the number of lungs that could be used. But you can see that is that little thing took a long time and was very difficult. I could not do it myself. It took a bunch of lawyers to do it, to figure it out, to really get policy changed, to say we're not doing anything bad. Nobody conceived of the fact that you would take an organ and repair it. The first EVLP in the United States was done in Toronto. There was a patient in Chicago that needed ex vivo lung repair, and the lungs were in another state, and this wasn't approved or known of in the US yet. So they called and asked me, can we do it in Toronto and bring it to Chicago? And we got compassionate approval. We flew the lungs to Toronto, repaired them at TGH, flew them to Chicago, where they did a successful transplant. Again, crossing the borders and all of that, people never anticipated that was going to be done. So these kind of policies are going to have to be changed.
Okay.
Great.
Thank you very much for that very educational and fantastic talk. On behalf of the Woodsworth Association, we would just like to convey our sincere gratitude for this.
Thanks very much.
In addition to Dr. Keshavjee, I would also like to thank all of you for coming out. If this is one of your first times at our events, we would love to have you at our future events as well. I would also like to take this opportunity to thank Carolyn, who is our Woodsworth Alumni and Development Officer at Woodsworth, as well as Benjamin for the tech support. Thank you for helping us run this event very well. And before saying goodbye, I would like to say that we will be sending out a short survey after this event. If you could please fill it out, we would really like to hear your feedback so we can better tailor our future events. So that concludes our evening. Thank you all for coming. We have some food and drinks at the back if you'd like to mingle. And thank you very much to our online guests. And good night.
I
really thought it was incredible. I'm mind blown.